Synthetic biology is a design-driven discipline centered on engineering novel biological functions through the discovery, characterization, and repurposing of molecular parts. I will highlight the applications of synthetic biology in the development of DNA and RNA-based vaccines. To make DNA vaccines for coronavirus, single-stranded RNA of the SARS-CoV-2 spike protein is extracted, synthesized into double-strand DNA and cloned into a plasmid. The plasmid is then injected intramuscularly in conjunction with an electroporation device to facilitate uptake. Within the muscles, myocytes take up the plasmid and express the protein of interest. This leads to either CD8+ T cell activation through MHC class I or B cell activation. Antigen-presenting cells like macrophages will endocytose spike proteins from necrotizing myocytes, overall leading to the recruitment of multiple immune subsets. Benefits of nucleic acid vaccines include almost any protein epitope to be targeted, their relatively high thermostability, and their rapidity of design and streamlined manufacturing process. But what makes RNA vaccines more efficient and how do they work? RNA vaccines share many advantages of DNA vaccines, such as rapid design and ease of manufacturing. But they do not need electroporation since they must only cross one lipid bilayer for cytoplasmic translation to produce antigens. mRNA vaccines can be manufactured faster and more cost-effectively than conventional protein-based vaccines. Researchers select mRNA that encodes the virus's spike protein. The mRNA is prepared by in vitro transcription where different components are engineered. Next, the mRNA is encapsulated with lipid nanoparticles. This layer protects the mRNA and raises a stronger immune response. The question you may ask is, where does synthetic biology come in play? Synthetic biology has allowed to increase the intracellular stability of vaccine RNA, reduce cytotoxicity, and enhance protein production. Here is a synthetically modified mRNA vaccine construct. Addition of 5-cap to RNA enhances translation and RNA stability by avoiding innate immunological recognition of uncapped 5-triphosphate RNAs. Addition of modular 5' and 3' untranslated regions identified high throughput screening to stabilize mRNAs and increase protein translation. Modification of sequence such as codon optimization has contributed to improved expression. Lastly, the length is important for translation and protection of mRNA molecule. Therefore, a 3' poly A tail is added. A study conducted in 2020 developing COVID-19 vaccines used RNA-based approach. Moderna mRNA 1273 is highly effective at preventing symptomatic COVID-19, but there is currently no information about its effects on the spread of SARS-CoV-2. Additional engineering of subsequent vaccine versions such as self-assembly mRNA vaccine may address some of the concerns with single-dose potency and long-term immunity. Unlike synthetic mRNA, self-amplifying mRNA is engineered to make more copies of itself. Once delivered into the cells, this means only a small initial dose is needed. To conclude, synthetic biology has allowed multiple vaccines to be developed against SARS-CoV-2 virus, then tested in clinical trials, granted emergency approval, and deployed within 10 months. Given the extraordinary progress of synthetic biology, it is sure to have a continued and lasting impact on the future of biomedical enterprise.